Okay, to start off our next session, we have Professor Nyasha Chambwe. Nyasha is an assistant professor in the Institute for Molecular Medicine at the Feinstein Institute in New York City. She received her PhD in computational biology from Cornell Medicine, also in New York. And then after that, came and was a postdoc in Ilya's lab in 2014. So please join me in welcoming Nyasha.
Thank you, Dave, for that wonderful introduction. And thank you to everyone who put this event together. I'm very honored to be here today and very honored to have been invited to say a few words. I think this is probably, I told everyone, I think this is the most important talk I've ever given. It's also the most difficult talk to have planned to give because it was difficult to find the right balance between science and the personal story I wanted to tell about having been mentored by Ilya. And so the talk title I selected was The Enduring Influence of Ilya Shmulevich in Cancer Genomics. So my talk today is part anecdotal stories about having been in the lab and working with Ilya. He never liked us to say that we worked for him. What I took from that experience into how I lead my lab now, in essence. So I am an assistant professor now. I've had a lab now for about 3 years. I'm in New York. I'm at a I'm in a health system. I'm in a research institute that's embedded in a health system. So it's a little bit of a different environment. I'm also in a cancer center at the Cold Spring Harbor Lab Cancer Center. But I was here for about 5 years, and it was, in essence, the most pivotal 5 years of my career in science, but also my personal development as a person. And so I found this quote, and thank you, GPTs. The people who make the biggest impact in your life often arrive unexpectedly, and their significance is only realized in hindsight. So I met Ilya in around 2013-ish, I would say 2014. So other people have known Ilya for 25 years, 30 years, since he was born, or they've been married to him for, for a lifetime. But even in those 10 years, I feel like I felt like I knew him my whole life. And so I was wrapping up my PhD. So this is one of my papers from my PhD thesis, where we were studying diffuse large B-cell lymphomas. And it was the era of microarrays. And this was one of the platforms prior to the Illumina chip called the HELP assay with the microarray. And we had 140 samples, and that was quite wild. You know, it was a really decent sized cohort, enough to get a comment and profiled paper in Blood, in the journal Blood, sorry, not in written in Blood, literally. And so, you know, it was, this was when the very notion of using molecular characteristics like DNA methylation and grouping patients by a molecular readout like DNA methylation, identifying subgroups and with different characteristics, with different biology, and that this was prognostic in some way, was a fairly novel idea. We take it for granted now, I realize. And also realize that I'm old enough to be able to give a perspective on a field. Very, very sobering stuff. But it was around this time when I was writing my thesis when Ilya came to our institute to give a seminar. And my PhD advisor encouraged me to sign up to speak to the seminar speaker, and no one else signed up. So it was me and Ilya in a room with whiteboards. And you know what? That's where the magic happened with Ilya. And I essentially interviewed for my postdoc in that room. And in a seminar last night, one of the software engineers who developed Cancer Regulome Explorer was here. We had a little reunion, and I told him I joined the lab because of this browser. I think somebody else showed some of these visualizations. because we were sitting there and I said, you know, we had this cohort, we have 150 samples, but I'm feeling this frustration. We don't have enough data, we don't have enough patients, we don't have genomic data, we don't have expression data, and I just really want to do more. And then I was sitting in this room and this man shows up and he was literally, you can't see me, right? So he, you know, Ilya talks, gave seminars, literally jumping out of his feet, right? And then we have 10,000 samples in TCGA, and we have started to build this visualization, and we have so much data, we don't know what to do with it. So we're building new tools and new platforms, and we have so much, you know, you should come work with us. So I was like, that sounds great. So I came to Seattle, and I think shortly thereafter, like the first month I came, we had a lab retreat out in Carnation. I'd never really been camping before. And the first lab meeting we had, they said, oh, okay, so now everyone bring your tent, your sleeping bag, your everything. And then, yeah, we're ready to go. So, and then everybody got up to leave, and I was like, wait. Does everyone have tents and sleeping bags? Like, oh yeah, we all own those things. I was like, I just moved here from New York. I definitely do not own a tent or a sleeping bag or any of the above. And so Leslie, who was our lab, I wouldn't even call her lab manager, lab guru, found this park because it had these yurts. And then everyone was making fun of me because I needed a whole room with a shower and everything. But in the end, everybody stayed in the yurts, I must acknowledge. So I put this to say that when I got here, I discovered that there was a combination of the data, the platform, all the infrastructure, but also a very, very, very wonderful group of people. The words can't describe the kind of scientific environment that was here that enabled the kinds of discoveries and science you've been seeing all day, right? Software engineers, people with a physics background, people from all sorts of different countries in an open, collaborative environment with no judgment, because Ilya extended that to everyone, right? It was like, there were no stupid questions. If you had never heard of a PBN, you could lift your hand and ask, what is that? And somebody could explain it to you. You know, if you'd never heard of a cell, you could say, I don't know what a cell is. What does it do? I've come from physics. All viewpoints were welcomed, and we worked together as a team on all these different projects, and there was a lot of innovation. So I remember when the cloud pilot was announced, and they were building this thing in the cloud, and people were still asking, what is a cloud, and how are we going to use it, and why is that important? And the fact that we now routinely do all our computation in the cloud, that is one of the things, upon reflection, I was thinking about how Ilya is so thinking and including taking input from all the people in the group, thinking and seeing the vision that ultimately we would be doing this large scale compute in the cloud was so at the forefront, right? Most institutions now don't even invest in an on-prem cluster, right? It's just a fact. People are just moving to cloud-based solutions for everything. And how the ISB CGC resource has matured, I routinely hear people asking me, oh, like, have you heard of the ISB CGC? I was like, oh yeah, I'm familiar with it. I may have used it a time or two. And so I'm very proud to have come from this lab where this work was done, and also to have been one of the early testers and users of this technology. So when I was wrapping up my dissertation, one of the papers that Ilya and other leaders in the cancer perspective commentaries that they published was this idea of a pan-cancer analysis, or the pan-cancer idea. This was not something that had been done before. The idea that you could merge data from multiple cancer types and analyze it jointly was novel. It was, in fact, I would suggest that it was radical to some degree. So this commentary was published, I think, 2012. And so all the work that by the time I came to Seattle was the idea that we would combine all of these data modalities, as Teo alluded to, and analyze them from a thematic point of view was already underway. This was organized from high level by sort of groups like cell of origin. So tumors that had a likely similar cell of origin were analyzed together, sort of squamous cell, putative cell of origin, or oncogenic process perspective, or key cancer-related pathways. And so I was, our group, the lab was involved in many of these initiatives. And so a summary of the pan-cancer effort from the Shmulevich lab perspective was that in 2018, when all the final papers were released, we were named authors on 11 out of the 27 papers. So I'd say that was an outstanding contribution. And then most importantly, some of the foundational work across the network was done on the computational resources and tools and that were built in the lab that Ilya had the vision to develop ahead of time, right? So the CGC platform, for example, was used for the baseline calls for germline variant calling. And then, of course, the jewel in the crown, so to speak, the immune working group and CIR, what is now iAtlas, all the data fed into that platform, and all the data is still available now. And you can do secondary and downstream analysis from there, and have continued to build on those tools as time has gone on. So not only in reflecting on all of this, on this work and this period at this time, because at least I came in after the initial TCGA was done, but towards the end, that not only was Ilya there at the beginning, he was there at the end, and then he was also there, shepherded a phase where they built the kind of infrastructure that would create sustainable cancer genomics, cancer accessibility to this data, and reusability of this data for other users as well as for us as well in other projects and beyond going forward. So by way of vignette, I worked primarily on the pathway working group for DNA damage repair. This was strategic from the point of view that this was a key pathway of interest for us. It's a key pathway that's important therapeutically, but it was also related to some of the other projects we were working on in the lab. In this project, locally, one of our collaborators, Raymond at UW, was one of the co-leads of the project. And then from our group, I worked with Theo on this project as well. Quick summary of this project was to focus in on the DNA damage repair pathway in terms of loss of function events in cancer in this pathway, either through mutation or epigenetic silencing or deletion events, to ask what are the functional consequences of loss of DDR functionality, and what molecular data can we use to estimate functional consequences of DDR loss, and to determine if they have clinical consequences or if they can determine clinical outcomes. One of the, which is now kind of one of those things about TCGA that you always think about, is that it wasn't clear exactly what are it wasn't set in stone, or we all know that they are DNA repaired genes, but the pathway curation ended up taking, I think, close to a year. You'd think that, oh yeah, this pathway is really important in cancer. We know what the DDR genes, but there was a long discussion, synthesis, expert consultation to come up with the 276 genes that are considered DDR genes. And then there was further refinement of a core set of DDR genes. It turns out that I refer people to this paper more often than not. It's now one of my most cited papers, and perhaps not that surprising. But I also then use this gene set all the time. So another enduring legacy of these resources we built over time. We looked at which of these types of alterations are the most frequent across cancer types, across sub-pathways of DDR sub-pathways, and looked at these interesting patterns. One particularly clinically relevant DDR signature is homologous recombination deficiency because it has implications for treatment. So we looked at how homologous recombination deficiency by copy number related, from copy number derived scores impacts outcomes. Primarily, we saw similar patterns across most cancer types, although, for example, here, whereas in other cancer types, a low HRD score was related to favorable outcome, this pattern was reversed in GBM. And we started to see patterns like that and explore that further in different tumor types. So this was the kind of power or landscape you could have in a pan-cancer analysis where you'd look at how global patterns were the same or different across different tumor types, and then this opened up investigation in tumor type specific ways or follow-up studies in different cohorts. So I think that as an enduring legacy, the Pan-Cancer Atlas phase was fairly successful. These resources are still used routinely. And for example, people will, as a starting point, if they're looking at a driver analysis of any kind, they'll look here first and then go from there. If they are looking for immune deconvolution, any starting point now in many cancer studies, or any reviewer will ask, have you compared to this 2018 set of findings, for example? But what I was also thinking about was how we ourselves as a group in Ilya's lab used these resources we developed in our own projects and in other collaborations, and how we leverage these for our local collaborations. So I referred to a project with Ray Monat at UW, for example, where we had a PO1 project looking at the molecular determinants of cancer therapeutic response. So our collaborators here at UW were primarily interested in DNA damage repair and its impact on therapeutic response. And over the course of that grant, we published many papers with lab members, including and up to this past year. And for example, here they were looking at formaldehyde sensitivity screens in cell lines, but those cell lines hadn't been genomically profiled. But we had those, some of those cell lines already profiled in public data sets in CCLE and GDSC, and that data was already available through ISB-CGC platform. So it was fairly easy and standard and routine for us collaborating through these types of projects to then join our local cancer cell line screening data with our large-scale public data so that we could interpret the findings from those sort of drug screening or drug sensitivity cell line screens. Similarly, Ilya was not always out there in the world doing stuff. We had other collaborations as well down the street. Chris Kemp, our collaborator, is here from the Fred Hutch, where we also had a year one award with them for 5 years initially, and apparently was just renewed and has continued over the course of the past, say, 10 years as well, looking at essentially looking to move from a phase of molecular characterization to identifying therapeutic targets, but leveraging all the lessons learned from TCGA and molecular characterization to the next phase of identifying therapeutic targets. And in here, for example, we're particularly interested in, for example, synthetic lethality mechanisms. And so together with Baha and Guangrang, who are still now in the lab, we leveraged ISB-CGC to build computational pipelines using cloud-based notebooks, leveraging public data to build computational pipelines to predict synthetic lethal interactions, which would then leverage through those CTD squared collaborations to validate and compare with the lab-based validation data sets. So around the time when we had wrapped up these projects, I started thinking about what was next. So similar to that transition time after my PhD, I started having long coffee conversations with Ilya about where I was going next. And if for some of you who remember, when I was at ISB at that time, we used to have these lunch brown bags. And so there were things being published around that time about the lack of representation of non-Europeans in different genetic studies and the impacts on precision medicine broadly. And I started to ask myself questions about what the impacts would be on precision oncology specifically, for example. So for example, here in GWAS, it was showing that even after the alarm was initially raised in 2011, there was very little change from 2009 to 2016. So although non-European representation changed, increased, there was still not as proportional an increase as you would have liked given the lack of representation over time. Similarly, we've known and continue to know that for decades, racial and ethnic disparities in cancer have persisted, exist, and continue to persist, where we see, for example, that people who self-identify as non-Hispanic Black have the highest incidence and mortality due to cancer in the US. And we have global variation in cancer incidence and mortality, usually attributed to, say, social, healthcare, and access issues. However, it's not that simple. So I started to think about, for example, how, to what extent do we know, for example, how the ancestry effects, what ancestry effects in cancer are like, and how representational are the cancer genomics cohorts, and what impact does that have on what we know about oncogenic drivers and the impact that would have on therapeutic development. So I wrote up this very nice one-pager about the project that I wanted to do, and I brought it to Ilya. And I thought for sure I'd get approval to do this. We have the data, we have access to it. I'm gonna write a data access request. I'm gonna use TCGA data, I'm gonna use the germline data, and I'm gonna do my project. And Ilya says, well, this is brilliant, Nyasha. I applaud you, this is a great idea. However, there is a TCGA working group that just started proposing to do the exact same thing. Let me make a few calls. So he made a few calls and I joined this working group. It was called the Ancestry Informative Markers Working Group. And essentially that was what the group did, where we similarly used the same TCGA data, the germline data, used it to infer genetic ancestry, and then asked the simple question, the first question: if we consider genetic ancestry, do we see ancestry-associated molecular effects in cancer? And the answer is yes, but, or yes, and this is not the right cohort to do these types of studies. So in summary, the TCGA cohort by genetically inferred ancestry is 83% European ancestry, with approximately 6% respectively East Asian and African ancestry. Even with that limited representation, we still showed that you'd see different, for example, mutation burden, ancestry-associated mutation burden between African and European ancestry individuals, and that you see, for example, depletion in particular types of mutation in specific cancer types between different ancestry groups. So here, kidney cancer, you see, which are known, VHL and PRBM1 mutations are depleted in individuals of African ancestry. We also saw ancestry-associated effects in expression, methylation, microRNA expression, and then we tested whether those molecular events are being driven genetically. So one of the prevailing arguments in disparities research is that these effects are not biological. They're simply a reflection of environmental effects or social effects that relate to, say, poverty or social ills or lack of healthcare access. But through this TCGA data, we're able to show that at least in part, that there are ancestry-related or population genetic variation at play in explaining some of the different molecular characteristics we see in cancer phenotypes. And so my lab now focuses on cancer health disparities, looking at ancestry-related genetics and biology, whilst considering the societal level cultural frameworks, neighborhood and social economic conditions, as well as institutional and environmental factors where possible. And so we hope to be able to determine biological and genetic factors that contribute to disparities whilst using race, ethnicity, and ancestry as a lens in performing multi-omic profiling studies in cohorts that are as diverse as possible, and to be able to determine these features that contribute to disparities so that we can build better models for risk assessment, prognostication, so that we can have treatment outcomes that are more equitable for all. And to finish, I just want to say that this quote I thought was apropos. I don't know how, if I would have got to here if I hadn't been here, but being here certainly helped me to get here where I am now, there, here, there, everywhere. But Ilya's teaching, mentoring, enabling certainly played a large role in getting me to where I am now. And I certainly hope that now that I'm responsible for some other students, that his legacy lives on in some way, in the way that I transfer what I learned from him to others. So thank you so much.